Hello students, this is Faizan Mirza. We are discussing the topic of selection and evolution. The subtopic that we are discussing right now is basically population. So what is a population? Population actually refers to a group of individuals of a certain species which are living there at a certain time in a certain region. So you can see that, for example, if this was a whole lawn and this was a snail population growing here, so the entire lawn area, the entire this vegetation would have several snails, and all these snails would be having different kind of genotypes and different kind of allele combinations, which will be representing the gene pool of these snails living here, and they would be constantly reproducing and they would be constantly reshuffling their existing alleles. Now let's suppose a hedge forms here. Now when the hedge forms here, this population of this snail cannot migrate there, and they cannot migrate here, and even if migration occurs, a limited migration can occur. So this is a semi-open population of these snails in this vegetable patch. So they will keep on reproducing with the you can see that the genetic combination that they have are the ones which they will be passing on to their offspring. Whereas since they cannot pass on there, and these ones cannot come here very you can say more in number, so the genes which are the genetic combinations of this population of snails and this combination of this population of snails is less likely to intermix together. Now let's suppose over the time here an open population of snails form because of a flower bed. Here you can see flower bed is having a larger area for these snails to move. So the snails can move and there can be migration in these populations. So this population and this population of snails can actually they can intermingle, and they can reshuffle their alleles by reproducing. Now let's suppose over time something else occurs as well. So let's suppose here that the roads are laid down in this vegetable patch, and in this lawn, for example, a road is laid here, and then there is a circular road here, and then some more interconnecting roads. So this population of snail, which initially could intermingle everywhere, got trapped here. So this is a closed population. It cannot successfully cross this road and go and mingle with the other population. The snails on this traffic island here on the flower bed here, they represent a closed population. So they would just reproduce together. They will pass on the alleles which they have. This is a semi-open population. This is an open population which can freely migrate there. This is also an open population here, and this is a semi-open population which can probably migrate there, and probably not a limited number of snails would only cross this particular hedge. So this basically actually is the idea that we need to keep regarding population, that since the populations cannot intermingle of a certain, although they were all initially the same species, since they cannot intermingle, so they cannot reshuffle all the alleles which were initially present, and they were able to do that. So populations, subpopulations are made within the same population, and the allele frequency of this particular population of snail and this population of snail and this population of snail would differ. So the offsprings here will not represent the entire same allele frequency which is seen here or which is seen here. This is the basic concept that we are to discuss here. This brings us to genetic drift and the founder's effect. What is genetic drift? Genetic drift is basically a change in allele frequency that occurs by chance because only some organism of each generation are able to reproduce. And when this occurs, there is a loss of genetic variation over time, and the new population which is established because only some of them got able to reproduce by a very small number of individuals from a larger population, this is not genetically representative of the actual population or the natural population that existed before that. So the gene pool in this case will be limited. For example, you can take some examples of Ashkenazi Jews, or probably the French Canadians, or the Martha's Vineyard of deaf people. These are all the isolated populations in which this genetic drift have occurred. And probably you can just refer to Dan Brown's novel of Inferno. This is an example that I give in my class. Anyways, I would skip this for now. But Ashkenazi Jews and French Canadians, these two populations, you can just surf the net and you can see that these individuals, they are forming a closed population. They do not intermingle with other human populations of other religions. The Ashkenazi Jews or French Canadians who are non-French Canadians, they do not marry with them. So their offspring, they suffer from a lot of genetic diseases which are uncommon in the remaining human human population, because the most of the genetic diseases are recessive conditions. So the likelihood of the homozygous recessive condition prevailing in the offsprings become more common when the same population ends up reproducing again and again and again without intermingling with the larger population. The Martha's vineyard of deaf people is exactly the same kind of the example here. This Martha's vineyard of deaf people is actually a village in Massachusetts, and they all people there are deaf. They do not mingle with the other population they're surrounding, and they only marry within their own population. So when genetic drift is occurring in a when this is occurring, this entire condition where only a few organisms are able to reproduce, when this is occurring in a recently isolated population, a recently isolated small population, this is called as the founder's effect. The same example that we were discussing in the previous slide, that the snails got caught in a closed island on the surrounded by the road. So the founder's effect would be seen there. The allele frequency of their offspring will not be representative of the true alleles or the gene pool of the original population. Next, here we are discussing Hardy-Weinberg's principle, which is applied for these kind of situations. We use two equations here: p plus q is equals to 1 and p square plus 2pq plus q square is equals to 1. What is q square? Q square represents the homozygous recessive in a given population, and the under root of that q square represents a single recessive allele. P square represents the homozygous dominant in a population, and under root of that p square represents a single dominant allele. P and Q, when multiplied by 2, it represents the heterozygous dominant in the population. So we use this kind of these two equations to just equate and find out the frequency of a certain allele in a particular population. Hardy-Weinberg principle is applicable in most cases, but it is not applicable when the mating is non-random or when migration into and out of population is occurring in a given region, or probably when the reproduction is asexual, or when mutation has occurred, or when the haploid condition is there, or even when there's a significant selection pressure against a particular allele. Now, taking an example of how to use Hardy-Weinberg principle. So the example given to you here actually tells you that this PTC is a chemical which can be tasted by individuals in a population because of a dominant allele capital T. So if dominant allele T is present there, the people are called as tasters. So tasters are capital T or heterozygous capital T small t, whereas non-tasters who cannot taste this chemical PTC are small t homozygous recessive. Now, in a local population, it is already mentioned that 200 people in Western Europe of these 130, which means 65% were tasters, and 70, which is 35%, were non-tasters. So we can see that 0.65 and 0.35 can be worked out for the particular percentages given to us. 0.35 here represents recessive condition, which is q square, and 0.65 is the dominant, which is the homozygous as well as heterozygous condition. So if we just take out the square root of q square, so q square, the value is 0.35. If we take out this square root of this particular value, which is 0.35, our answer comes out to be 0.59. So we know that 0.59 is the frequency of the non-tasters in this particular population. Let's take another example. Suppose the incidence of homozygous recessive trait is 1%. Calculate the percentage of homozygous dominant and the heterozygous dominant in a population. So we know that 1% is given to us. This means the homozygous recessive is 1%, which means 1 upon 100, which is 0.01. So homozygous recessive condition, 1% is actually your q square. So what do? You work out the q value here. The q comes out to be 0.1. Once you have q, this is the single homozygous recessive allele. Now you apply p plus q is equals to 1. You just equate the value and you just rearrange the formula for p. So p is equals to 1 minus 0.1, and p comes out to be 0.9. So this p represents a single dominant allele here. So once you have the single dominant allele, to find out the frequency of how much it is there, we just turn it into a square. We use p square here. So 0.9, so you just square this value, turns out to be 0.81, which means that 81% population here is homozygous dominant, whereas 1% was just homozygous recessive. Now, to work out the heterozygous condition, what do? We can use the formula which gives us 2pq. So using 2pq, we just substitute the values of p and q, 0.9 and 0.1 here. Multiplying it with 2, we come out with the value that our 2pq value comes out to be 0.18. 0.18 confers to 18% in a population. So 18% of population of heterozygous. So 2pq, since 2pq represents heterozygous dominant, so 18% is our answer, or 0.18, the frequency is our answer. Do not go on to solve this mathematically. This is a very common error that the students do, that they just solve and they come out with the value for PQ, which is actually wrong. The other way to just check your answer is you have a homozygous dominant in 81% in the population, and you have 1% which is homozygous recessive in population. If you add 81% with 1%, this gives to you 82%. So 82% of individuals are homozygous dominant and homozygous recessive. You just subtract it from 100%, so that again gives you 18%. So that's another way of rechecking your answer, that how that whether you worked out the correct answers for the three homozygous recessive, homozygous dominant, and heterozygous dominant or not. Lastly, here we'll be discussing the process of artificial selection. And when we're discussing artificial selection, why artificial selection is actually the same thing, which is selection pressure, which we are exerting as human beings. So natural selection is something that the nature is doing. Artificial selection is something that we are doing. So it basically is just selection pressure being exerted on a certain kind of organism to just make sure that what we want from the population of a certain organism that can be produced. So artificial selection of plants and animals have occurred throughout the human history, and individuals with desirable features and desirable traits are chosen to so that they can be interbred, and the resulting population will have the desired traits. Now, the desired alleles will be passed on to the offspring successfully if this is done effectively. And as I already mentioned, that here the selection pressure is being exerted by the human beings. The desirable traits for in a particular animal that one can look for in when the person, when we are involved in farming, is an example of cattle farming. We look for docility, meat, milk, color of hide of particular organism. Docility means how easily the animal is to control. The animal is not very aggressive. So these are the traits that are favorable traits in farming. So for this, progeny testing is carried out. Progeny testing means that you test the offspring for the production of that particular trait, or whether the trait is present or not. So in progeny testing, the most difficult one is to test the progeny for milk production, because milk production is a sex limited trait. What do you mean by sex limited trait? Means that it is related to a certain gender and not the other gender. So genes for this come from cow and bull both. But bull cannot be directly assessed for the milk production, since the bulls do not have the mammary glands for, or you can say effective mammary glands for production of milk. So you cannot assess a bull for its milk quality, although the alleles will be there in bull. So the offspring will be having the allele combination from the mother cow and from the father bull. But the thing is that since we cannot test a bull for that particular trait, it's difficult here. So what do? We have to go for the progeny testing. The bull's offsprings are then observed for the quality of milk that they are producing, the quantity of milk they are testing. This is called as progeny testing. This is not an easy process. Why? Because number one, it's time consuming, as the cows cannot produce milk till a certain age, so they have to grow to that certain age. Another reason is that the progeny might be male, so it may grow on to become male, just like the father bull. And by the time the bull is assessed for its sex limited trait, the bull would have reproduced and would have many offsprings by then. So this is actually a challenge for artificial selection, which is being carried out in cattle farming. Similar processes are carried out for crop improvement, and in crop improvement, desirable traits and desirable features which one look for, these include increased yield and pest resistance. These are the common features, and as well as some disease resistance or vigorous growth in the crops and the quality of the product. For example, if it's a food, so how good the crop is, as well as the uniformity in the growth, so that the entire field can be harvested at once. So wheat and rice, over the years, they have been made disease resistant by passing these favorable alleles from other varieties of these species which are found in wild. And the dwarf varieties of plants can grow tall upon getting the gibberellins. We already have discussed gibberellin in one of the previous topics. The problems here is there are certain problems here when we are discussing this. So inbreeding, how is this inbreeding carried out? Inbreeding means that offspring produced from the breeding of individuals which are closely related. This kind of breeding is called as inbreeding, and this over the time leads to homozygosity, and homozygosity initially is very important for developing these conditions, but over the time it leads to the decline in. So this homozygosity leads to increased chance of passing the recessive trait as well. So this is a challenge here because we know that recessive traits, mostly they are the ones which are the basis of genetic diseases. So homozygosity although can favor this, but over the time it passes the recessive disorders to the offsprings, and this reduces the biological fitness of a population and it affects the crop adversely. So this is called as inbreeding depression. Now to just combat this inbreeding depression, over generations, what's done every 2-3 generations later, outcrossing is carried out. What is outcrossing? Outcrossing means that this is actually done to avoid inbreeding, and this is inbreeding depression. This is also called as inbreeding avoidance. How is this done? How is this carried out? Inbreeding and hybridization is carried out using much fit varieties from the population which are existing in the wild. They are crossed together. So if you have a homozygous population here, you just cross it with a more fit population from the wild. This is called as outcrossing. This promotes heterozygosity, and this inbreeding and hybridization together, they produce a more fit varieties of population over time. For example, maize, more you can say the fit populations of maize have been produced, and same with wheat and rice and cotton. They all have been produced over the years with the same process. So every year, every few years, inbreeding leads to homozygosity, which leads to a reduced biological fitness, and then you have to perform outcrossing to make sure that the biological fitness of the population stays high, and the inbreeding avoidance can be attributed to that. That's it for now. Thank you so much.